Hi everyone, today we'll be solving Cambridge International AS Level Biology 9700 Paper 2-1 May June 2023. Figure 1.1 is a transmission electron micrograph of a cell from the stem of sago pondweed, Stacania pectinata. We can see the label parts A, B, C, D, E, F, G, H. EFG, and cell wall and airspace is labeled. State the evidence from figure 1.1 that shows that the cell is from the stem of S. pectinata and not from the mesophyll of a leaf. We can see in the diagram there is no chloroplast. If it would be from a mesophyll, then there would be chloroplast. So we would say no chloroplast present. Complete each row in table 1.1 to identify a cell structure shown in figure 1.1 that carries out the function listed. Gas exchange. Name of the cell structure that does gas exchange. So first of all, let's label the structures that we see in this particular diagram. First of all, the structure A is the cell membrane. Structure B is the nuclear envelope or nucleus. Structure C is the nucleolus. Structure D could be starch molecule. Structure E is definitely mitochondria. F is it's like a tonoplast or a vacuolar membrane. And structure G shows us either smooth endoplasmic reticulum or rough endoplasmic reticulum. So we'll say endoplasmic reticulum. D could be starch molecule. Now, here it says function and name of the cell structure. Now, we know the cell structure that does gas exchange is cell membrane. Production of subunits of ribosome is definitely nucleolus. Active transport of ions is definitely cell membrane. Okay, we'll have to mention the word cell surface membrane because there are many more membranes present within the diagram. And then aerobic respiration is definitely mitochondria. So, cell membrane is labeled with A, cell surface membrane, and nucleolus is labeled with C, and mitochondria is labeled with E. Plant vacuoles overlap when vesicles fuse together. The vacuoles increase in size as more vesicles fuse. Figure 1.2 shows the movement of vesicles within a plant cell during the development of a vacuole. So, in the diagram, we can see fusion of vesicles. Now, the cell surface membrane is doing endocytosis at point X. Rough endoplasmic reticulum is synthesizing and packaging polypeptides or protein, which is destined for the Golgi body. And from the Golgi body, so fusion of vesicle occurs here, and vesicle fuses with the vacuole as well. Name the process that is occurring at X. So, process occurring at X is endocytosis, or we can say pinocytosis. The term endocytosis is referred as cell eating, whereas the term pinocytosis is referred to as cell drinking. Some of the vesicles formed by the Golgi body pass to the vacuole. These vesicles contain proteins that have been folded correctly and some that not have folded into their correct shapes. The proteins that have not folded correctly pass to the vacuole where they're broken down. Folding correctly of a protein is important because it gives it active site. Now, without an active site, a protein is not usable. Explain how proteins that have not folded correctly are broken down into vacuole. So, vacuole may attach itself with lysozyme, which contains lysosome, and as a result, hydrolysis will occur. So, the answer can be written: proteins that did not fold correctly are broken down by hydrolysis. The action of protease enzyme on the polypeptide breaks down the peptide bonds and form amino acids. Small vacuoles in S. pectinata may have roles similar to lysosomes in animal cells. Describe the roles of lysosomes in animal cells in defense against pathogens. Now, we know that lysosome contains hydrolytic enzymes. So the answer can be written: phagosomes fuses with lysosome to form phagolysosomes. So this is the hybrid of a phagosome fusing with a lysosome. Once the lysosome releases its enzyme, so lysosome contains lysozyme enzyme, which then breaks down or digests the pathogen/bacteria. Glycogen and cellulose are polymers. Figure 2.1 shows small representative region of glycogen molecules and a cellulose molecules. We can see here 1,6 glycosidic bond, 1, and the 6th position glycosidic bond. And this one represents 1,4 glycosidic bond, whereas in cellulose we see 1,4 glycosidic bond as well. This is the similarity between both glycogen and cellulose. However, the cellulose molecule, the glucose molecules are 180 degree inverted. The 6th position carbons are 180 degree inverted. The question says describe three ways visible in figure 2.1 in which the molecule of glycogen differs from the molecule of cellulose. So the answer can be written as follows. Glycogen is branched, versus the cellulose molecules which is unbranched. Glycogen monomers are made of alpha glucose, whereas cellulose molecules are made from beta glucose. Glycogen contains 1,6-glycosidic bond as well as 1,4-glycosidic bond, whereas cellulose contains only 1,4-glycosidic bond. And on the fourth point, and an additional extra point we can write, 1,4-glycosidic bonds are not rotated 180 degrees or they are not inverted, whereas cellulose molecules, their 1,4-glycosidic bonds are alternate or inverted. Now guys, because our answer says that we have to write how glycogen differs, so we do not have to write the portion for the cellulose because it says describe and doesn't say compare. Glycogen is found in the form of granules in mammalian liver and muscle cells. Figure 2.2 is a diagram of a part of molecule of glycogen isolated from glycogen granule. So we can see glycogen that is isolated from a glycogen granule and a small part is shown. Explain how the structure of glycogen is related to its function in cells. Now, few things that we need to notice is highly branched. So highly branched means it will be easily broken down in hydrolysis, where it will produce many ends. So the answer can be written as follows. Glycogen is a store of energy and it is highly branching, so faster release of glucose is possible whenever needed. It is compact, so stores a lot of glucose in small space. Now this makes it 3 points, so we'll get the full marks. However, there are 2 more added points which you should only write if you cannot write the first 2 points or if you forget them, which is it is insoluble, so does not change water potential. Now write any 3 points to get the full marks. However, it is not a good idea to write many points because if we write wrong answers in any one of the points, then we may lose a mark in spite of having all 3 corrects. So better to write as little as point possible according to our best knowledge, and only write more if we know that our answers that are given are correct but may not cover all the points. Then you can write a little bit more. Explain how the arrangement of cellulose molecules in plant cell walls is related to their function. So the answer can be written as follows. Cellulose molecules are unbranched polymer, so they can be arranged in parallel. Cellulose molecules contains many hydrogen bonds between the cellulose molecules. This allows them to form microfibrils, and the hydrogen bonds give them high tensile strength to form microfibrils. Now guys, 5 points are written here so that you can, if you forget any one of the 2 points, then you can still write a full answer. All right, so choose to write only between 3 to 4 points during your answer. Figure 3.1 shows some fruits, grapes of the grapevine, which is vinifera. Sucrose is transported in the phloem of the grapevine to the fruits. In the fruits, sucrose is hydrolyzed by the enzyme sucrase, which is found in cell walls. The glucose and fructose produced by the hydrolysis of sucrose pass through the membrane proteins known as hexose transporters into the cytoplasm of the fruit cells. Now, hexose means 6-carbon sugar. So as you can see, the name suggests hexose transporter means 6-carbon sugar transporter. Now, when sucrose is hydrolyzed, sucrose is made out of glucose and fructose. Both are hexose sugar, 6-carbon containing sugar. State why membrane proteins are required for the movement of molecules such as glucose across the cell surface membrane into cells. Glucose is a polar molecule, and cell membrane by its nature, the phospholipid bilayer, is nonpolar. So a transport protein allows it creates a channel where molecules like glucose can enter through the channel. So we can write sucrose is a polar/hydrophilic molecule. So it cannot pass through the phospholipid bilayer in cell surface membrane. Researchers investigated one type of hexose transporter known as VVHT1, which is found in the fruit cells of V. vinifera. They used a mutant strain of yeast that has very few hexose transporter in its cell surface membrane to investigate the properties of VVHT1. The researcher inserted the molecules of VVHT1 into the cell surface membrane of the mutant strain of yeast. Equal volumes of mutant yeast cells with VVHT1 were kept in 8 different concentrations of glucose solution. Now, the rate of uptake of glucose by the yeast cell in each solution was determined. All the solutions were kept at the same temperature and pH. The results are shown in figure 3.2. So, in general understanding here, the VVHT1 is a transporter, meaning it is a channel protein. Its purpose is to allow glucose or hexose sugar to pass through the cell membrane. Now, the type of yeast, all right, that we are analyzing here, we inserted the VVHT1 in its cell membrane. So now we want to see how much glucose is uptaken by the yeast cell in each solution, all right, so in each concentration. So if we increase concentration, does the rate of uptake increase or not? The graph shows concentration of glucose micromole per dm cube. As we increase the concentration of glucose in micromole per dm cube, the rate of uptake of glucose in micromole per minute per gram increased. However, it plateaus at some point. So what does this indicate? This indicates that all the transporter proteins, the VVHT1 transporter, are saturated. Okay, that's one information there. And the other information is, as we increase the concentration, all right, this particular VVHT1, it's a channel protein that only carries the glucose by means of facilitated diffusion, because as the concentration increased, the rate of uptake also increased. Now, the question says the researchers concluded that VVHT1 is responsible for facilitated diffusion of glucose into the cell. So our graph also gives us the same conclusion. Now the question says, explain how the results in figure 3.2 provide evidence to support this conclusion. So the answer can be written as follows. The rate of uptake becomes constant at concentration greater than 150 micromole per dm cube. So the protein carriers are working at their highest rate, or we can say they are saturated. The researchers thought that grapevines could be modified to have more hexose transporters to increase the size and quality of grapes. Explain why increasing the number of hexose transporter could be commercially important to growers of grapevines. If we put more hexose transporters, which will then bring in more glucose or more fructose, meaning we'll have sweeter grapes, which has a higher demand. And because there will be more glucose available for the grapes to grow and the plants to carry that particular glucose and transport it, so that particular plant will have a larger yield, means more profit. So the answer can be written: the grapes will be sweeter or will have improved taste, and they will also produce more yield, which will bring more profit for the farmers. Figure 3.3 is a diagram of protein in the cell surface membrane of a macrophage from a mouse. Now, you see, macrophage is a type of white blood cell which is similar to that of phagocyte. In simple terms, they engulf bacteria or dead cells and then digest them. Macrophages use these proteins in antigen presenting. Alright, so antigen presenting, basically they turn into APC or antigen presenting cells after they break, engulf and digest bacteria or viruses. Non-self antigens bind to the proteins and are involved in the activation of specific T lymphocyte during the immune response. So you can see the antigen binding region which is present here. State what is meant by non-self antigen. Non-self antigens are basically considered foreign proteins that stimulates an immune response in a particular organism. Some pathogens enter human cells. Macrophages partially digest these pathogens and present antigens to T lymphocyte during immune response. With reference to figure 3.3, how T lymphocytes respond to infection by a specific type of pathogen. When antigens are presented, these are forming APC or antigen presenting cells. The antigen presenting cells are then going to match with the type of T lymphocyte that are complementary to it. So this particular APC's goal will be to find complementary T lymphocyte, complementary T cell. Once the complementary T cell is found, then that T cell will get into the action of activating the B cell. The B cells are responsible for producing plasma cells, which will then produce antibodies. The antibodies will then, their action will be, again, the antibodies will fight off the pathogens directly by recognizing their antigen. So the answer can be written: clones of T lymphocytes have complementary receptors to an antigen. The selected T lymphocyte then undergo clonal expansion. The T helper cells then secrete cytokines to activate the B lymphocyte. Now it produces plasma and antibodies, and then the antibodies also kills off the particular pathogen. However, our answer is only asked to be written about T lymphocytes. So we will end this particular answer by saying T killer cells are also produced, which then binds to infected cells and destroys them by releasing toxins. The gene for the enzyme catalase is on chromosome 11 in humans. Explain the meaning of the term gene. So we can write the definition as follows. A gene is a sequence of nucleotides that is part of DNA and it codes for a polypeptide. Two enzymes, DNA polymerase and DNA ligase, are involved in the replication of DNA. Figure 4.1 shows the replication of part of human chromosome 11 by DNA polymerase. The arrow shows the direction of synthesis of new polypeptides by DNA polymerase. We can see DNA polymerase action. It is working from 3' direction towards 5'. It always works from 3' direction towards 5'. So this other segment is being the catalyzed in the reverse direction. So it will then move on from here and complete the rest of the job. Describe the role of DNA polymerase and DNA ligase in the replication of DNA. So the answer can be written as follows. DNA polymerase forms phosphodiester bonds between the adjacent nucleotides and growing strand. DNA polymerase also proofreads the new DNA polymer that is synthesized. DNA ligase joins the Okazaki fragments on the lagging strand. DNA ligase also forms the phosphodiester bonds between the fragments. State the name of the stage of interphase in cell cycle when DNA replication occurs. Now the phase is known as S phase or synthesis phase. Figure 4.2 is a diagram of chromosome 11 at metaphase of mitosis. We can see at the centromere, the spindle fibers are attached to the kinetochore, and B shows spindle fiber. Now, state the names and function of structures A and B. So, structure A is centromere, and the function is, it is the site for the attachment of spindle fibers, and also the site for the attachment of chromatids and spindle fibers. Structure B is spindle fibers, and the function for it is to orient the chromosome at the equator. Complete figure 4.1 to show what happens to the chromosome 11 in anaphase so that the daughter nuclei are genetically identical. We can see the cell surface membrane is labeled. The two centrioles are in opposite poles. So we are supposed to draw the spindle fibers. Now we are representing anaphase. In anaphase, separation already occurred. So we will have to draw our chromosome in this way because they are pulled towards the other ends. And from the place where they have centromere, we will draw centrioles that are connected to spindle fibers that are connected to centrioles. Figure 5.1 is a longitudinal section of a capillary in muscle tissue as viewed with a transmission electron microscope. So we can see the capillary that is carrying the red blood cells squeezing through, the capillary. State the evidence visible in figure 5.1 that identifies the cells inside the capillary as red blood cells. One of the distinct properties of red blood cell is its biconcave disc-like structure, and the other one is there is no nucleus. So the answer will be no nucleus, and some are biconcave shape. Explain how the structure of capillary wall is related to its function. Now you see, capillary wall is very flat and it's only one cell thick. This means substances can pass through it very easily by diffusion. Now, we will give the answer in this way. The capillary wall is very thin, only one cell thick, so provides a short diffusion distance for the passage and also provides endothelial pores for the passage of small molecules such as plasma, which turns into tissue fluid. So the answer can be written. Capillary wall is very thin, only one cell thick. This provides a short diffusion pathway. We can also add capillary wall contains endothelial pores for the passage of blood plasma, which turns into tissue fluid. Figure 5.2 is a diagram showing some of the events that occur as blood flows through a capillary in a respiring tissue. So in the place where respiring tissue is giving off carbon dioxide in high quantity, the carbon dioxide diffuses into the red blood cell. Now, the red blood cell consists of an enzyme known as carbonic anhydrase, which in turn with the help of water molecule, converts the carbon dioxide with water into carbonic acid. And then the carbonic acid dissociates into H plus ion and hydrogen carbonate ion. Now, the hydrogen carbonate ion is a big bulk which is then carried out of the red blood cell and into the blood plasma. And the formation of hydrogen ion. Now, this hydrogen ion also binds with the hemoglobin and forms HHB plus or hemoglobinic acid, which allows the release of oxygen much faster, and oxygen is then released into the surrounding respiring tissues. Now, in the lungs, just the opposite happens. An increase in respiration results an increase in carbon dioxide concentration in the blood and the release of more oxygen from the red blood cells to tissues. Explain how an increase in carbon dioxide in the blood lead to the release of more oxygen from red blood cells. So the answer can be written as follows. More carbon dioxide diffuses into red blood cells by the action of carbonic anhydrase in the red blood cell. CO2 combines with H2O to form carbonic acid. The H2CO3/carbonic acid then dissociates to form H plus ion and hydrogen carbonate ion. The hemoglobin then binds to the hydrogen ions and form hemoglobinic acid. This in turn releases more oxygen in respiring tissue. Fluoride ions are a constituent of blood plasma. The concentration of fluoride ion in the plasma of deoxygenated blood is between 2 to 4 mole per dm cube lower than the plasma of oxygenated blood. Explain why the concentration of fluoride ion in the blood plasma of deoxygenated blood is lower than in plasma of oxygenated blood. Fluoride ions are negatively charged ions. They are mainly needed to replace hydrogen carbonate ion lost from red blood cell. So the answer can be written as follows. In figure 6.1 is a diagram showing the passage of water through the tissues of a flowering plant from the soil to the atmosphere. The arrow shows the direction of water movement. We can see the water is moving into a leaf structure, moving into the spongy mesophyll cell, into the airspace as water vapor, and then leaving out through the stomata. Now, this creates a transpiration pool, which is then causing water to move into the root hair cells, and water then passes through plasmodesmata so with the symplastic pathway, then through the cell wall in apoplastic pathway into the xylem. And from the xylem, due to transpiration pool, it is pulled upwards. The structure label X is a part of the symplast pathway. State the name of the structure X. So the structure label X would be plasmodesma. Now, if you know the name plasmodesmata, remember that plasmodesmata is plural. So, since we are supposed to name only one of it, so we have to write plasmodesma. The structure labeled Y in the cell wall is a barrier to the apoplast pathway. State the name of the structure Y. So the structure Y is a waxy layer. It is a waxy layer which is known as Caspian strip. With reference to figure 6.1, complete the statement about the movement of water in the flowering plant. Water moves from the soil solution to the cytoplasm of root hair cells by. Now movement of water at this stage will be osmosis. Osmosis is the net movement of water through a partially permeable membrane from a region of high water potential to a region of low water potential. Water moves from the xylem into the root to the leaf by. So now here the process that is going to take place would be transpiration pool. Water moves from the mesophyll cells into the intercellular airspace by. Now from the mesophyll cell to the intercellular airspace would be simple evaporation because it is turning into water vapor. The water vapor moves from the intercellular airspace to the atmosphere outside the leaf by diffusion. Guys, that's all for this particular question paper. Thank you for watching this particular video. Guys, question paper videos like this will be uploaded regularly. So subscribe to the channel if these questions you find them familiar, or if you have a particular question paper in your mind which you want me to solve, maybe write down in the comments. Guys, best of luck for your exam and thank you for watching the video. And guys, please subscribe to support the channel. Bye bye.